Hello everyone, welcome to the third lecture of the series on the topic of neural control and coordination. So, this will be part 3 of the lecture and this is the last one in the series. So, as you know, we have been talking about in the previous 2 lectures how the nervous system actually functions, what is the structural unit of the nervous system, this is the neuron, how it generates the electrical impulse, how that is conducted from one neuron to the next. And in the second series, we looked at the structure of the brain, how overall the whole nervous system is organized and what are the division of labor in terms of how different parts of the nervous system perform different functions. So, today's topic is going to be focused on the sensory systems. So, as all of you are aware that sensory reception and processing is a very important function of our neural system. So, with this sensory perception, whatever changes that are taking place in our environment, we are able to sense that and we can detect these through the sense organs. So, all of you must be familiar with the various sense organs that we have, which are all dedicated, there are 5 of them, which are dedicated for detection of a particular sense. For example, let us look at them one by one. So, the first sense that all of you are aware of is vision, which is the ability to see. So, the vision is possible because of the sense organ, which is the human eye. So, all of you are familiar with the human eye and I will discuss in more details about it as we go on with the lecture. So, the next sense that you also must be aware of is the sense of smell. So, the sense organ in that case is the human nose, and the third sense that is also known as a chemical sense along with smell is the sense of taste, and the organ that we use for detection is the tongue, which is placed inside our mouth. Then the fourth sense that also is very important is the sense of touch, and for this we have the entire skin surface which has specific sensory organs, which I will describe later in the lecture, which are used for perceiving the sense of touch. For example, when somebody touches you, you are able to sense that that somebody has touched you. That pressure that is applied on your skin can be detected through this sensation of touch. Now, the fifth and the last is the sense of hearing, and all of you know that the sense organ that is devoted to this is the ear. So, let us begin with vision, which is the very important sense that all of you are aware of. So, which sensory system is the most important? I do not have to tell you that, you all know that vision is very important to all of us in our day to day lives, and sense organ that is devoted to this is the eye. So, the eye looks like this. So, it is like a spherical ball. If you remove the eyelids and if you take it out from the socket which is present in your skull, then you will see it is like a ball, a sphere. So, the front part of this ball has a little opening which is the lens, and it looks white more or less, except the part which you call as the iris, which has different colors. In most Indians it is either brown or black in color. In people from other countries it may be blue or green or various shades or combinations of these. So, in the next part of the slide I am going to show you what actually the inside of the eye looks like. So, what if you took a knife and you cut the eyeball just like this, right through the front portion, you cut it like this. What would you see? It would look something like this. So, this is the sphere cut in half and you are looking at the inner surface of the sphere. So, what you see is actually a hollow sphere, but this is not empty space here. There are 2 chambers actually. This is divided into a smaller part of the eye, which is the anterior chamber. This is called the aqueous chamber, and the larger part of the sphere which is behind the lens is called the vitreous chamber. Now, in between you have this structure which is called as the lens, which allows the light to be focused onto the retina. So, now I am going to tell you about each part of the eye one by one. So, the outermost layer is called as the sclera. So, this is the sclera. What you see as white colored in your eye, that is the sclera. But the sclera is made up of very dense connective tissue, and it is a tough structure, and its function is to protect this very delicate organ, the eye. Okay. So, in front, towards the front portion of your eye, where you have the lens, the sclera becomes very transparent. So, here it is no longer opaque and white like it is over here. So, this region of the sclera in the anterior part of your eye is called the cornea, and the reason it becomes transparent here is so that light can pass through, right. Then the next layer is called as the choroid. So, the cornea I just described to you, it is the transparent part of the sclera in the anterior part of the eye in front of the lens. Now, we come to the next layer. So, this hollow ball that we have, the eyeball, is lined by various layers. So, I am going from outside to the inside. So, the sclera was the outermost layer, then comes the choroid, which is the layer that is rich in blood vessels. So, it supplies nutrition and oxygen to all the cells within the eye, particularly the retina, which is the sensory organ, which is the only part that is composed of neurons within this whole eye. Okay. So, now this whole choroid layer, which has lot of blood vessels, extends into the anterior part of the eye, and here it becomes a little thick. So, this part is called as the ciliary body. Okay. And then it even extends a little bit further in front of the lens, and this is behind the cornea. So, remember there was the cornea which was the transparent layer. So, you can actually see through in this part, and that is why you can see the iris. So, the iris has various pigments which gives the color to the eye. Therefore, in case of this person who has a brown eye, this iris would be brown in color, right. Then the iris is actually not a continuous structure, it is interrupted in between. So, there is a hole in there. So, this hole is for the light to get in, and this hole is called as the pupil or the aperture of the eye, and this iris has a lot of muscles. So, this iris has concentric muscles that go around it like this, and when they constrict, the pupil becomes bigger. So, more light can get into the eye, and when you have other muscles which are called as radial muscles, when they contract, when the radial muscles will contract, then the eye becomes, the pupil becomes bigger, and when the circular muscles contract, the pupil will become smaller. So, that is the function of the iris and the pupil, to allow different amounts of light to get into the eye according to the situation. Then we come to the neural part of the eye, which is the retina. So, this is the part that has the ability to convert the light stimulus into electrical activity, because the retina is made up of neurons. It is the innermost layer of the eye. It contains certain specific neurons which are specialized to detect light and become electrically active as a result of it. These are called as photoreceptors. So, in the next part of the lecture, I am going to describe in detail the structure of the retina. So, again this is showing you the schematic of the inner view of the eye. You have cut the eye like this, and here the innermost layer, which is shown in red over here, is the retina, right. So, then let us see. So, here is the zoomed out view of the retina. So, if you take a cross section of the retina and you really zoom out, you can see that it has certain layers. So, it has 3 layers actually. So, you have the outermost layer, which is towards the outside, towards the choroid, is called as the photoreceptor layer. So, it has 2 kinds of cells. They are called as rod photoreceptors and they are called as cone photoreceptors. So, they are present throughout the retina. Now, the next layer of neurons which are making connections to this are known as the bipolar cells. So, you can easily see that the bipolar cells are called bipolar because they have one arm extending towards the photoreceptors and the other arm which is extending out towards the next layer of cells, which is called the ganglion cells. So, you have the photoreceptors. The photoreceptors are don't look typical like typical neurons. They have extensions, but you can't really tell what is an axon and what is a dendrite, because these are very small neurons. They do not need to send very long extensions out. So, they have very small processes. So, this is the receiving end, which is equivalent to the dendrite, and this is the output end, which is equivalent to the axon, and this makes a synapse with the bipolar cells. Then the bipolar cells will again have a process which is like the dendrite, and this process which is like the axon, which will make synapse with the dendrites of the next cell layer, which is called as the ganglion cell. So, the innermost cell layer within the retina, which is towards the vitreous, is actually the ganglion cell layer. Now, the ganglion cells look more like typical neurons, because they have short dendrites which make contact with the bipolar cells, and then they have long extensions which are the axons. So, these axons will travel along the inner surface of the eye from every portion. So, there are ganglion cells present all over the retina, and all their axons will travel on the inner surface of the eye, and they will come to a point and collect together in this bundle, and this bundle is known as the optic nerve, as I shall describe in a minute. But before that, let me tell you about how light is actually detected by the retina. So, this yellow line that I have drawn here shows the path of light. It will enter through the cornea, which is transparent, through the lens which will focus it onto the retina, and within the retina it passes through these two layers, the ganglion cell layer and the bipolar layer, and it finally reaches the photoreceptors, and it actually stimulates the photoreceptor layer. And then the photoreceptors can convert this light signal into electrical activity, and through the synapse it will convey it to the bipolar cell. The bipolar cell will then become electrically active and will be conducting this nerve impulse to the next cell, which is the ganglion cell. And as I said, the ganglion cell axons will all collect together and they form a bundle here, which comes out of the eye as the optic nerve. So, the optic nerve carries this information, this visual information from the eye to the part of our brain which can process this information. So, the blind spot is another region of the eye. So, the region where the retina is pierced by all the bundle of ganglion cell axons, it does not contain any photoreceptors, and hence that is called the blind spot in your eye. Okay. You are normally not aware of the blind spot because you can see everything in your visual field, that is in the area around you that is covered by your eye. Why are you not aware of your blind spot? The reason is that you have two eyes that are in front of your head, and so the visual field of each one overlaps to a certain extent with the other. So, the blind spot of one eye is covered by the retina photoreceptors present in the other eye. So, you are normally not aware when you have both eyes open of the blind spot. Only when you close one eye will you become aware of the blind spot that is present in your eye. Okay. So, now let us go and take a closer look at the photoreceptors themselves. So, as I said, the retina looks like this. This is the outermost layer, which is the photoreceptors, then you have the bipolar cells, and then you have the ganglion cells. Now, let us see what these photoreceptors look like and how do they function. So, the photoreceptors, as I described before, are rod and cone type of photoreceptors. Now, the name becomes very obvious from what it looks like, because you see this part, this is called the outer segment. This outer segment is rod shaped in rod photoreceptors, and it is kind of cone shaped in cone photoreceptors. Hence, the name rod and cones. But this is not the only difference between them. There are several other differences, and I will describe them one by one. So, as I said, light is capable of stimulating the photoreceptors. So, how does it stimulate the photoreceptors? This is because the photoreceptors have a very specific molecule in them. This is a protein which is called as rhodopsin in case of the rods and conopsin in case of the cones. And this protein is present in the membrane of the rod photoreceptors in this region. This outer segment part, which is rod shaped or cone shaped, has many discs of membrane. So, the discs have this rhodopsin or conopsin present within them, and it is usually associated with another small molecule called retinal. So, retinal is a derivative of vitamin A. So, vitamin A produces retinal in your body, and that retinal is associated with rhodopsin or conopsin, which is present in these stacks of membranes that are present in the outer segment of the photoreceptors. I will tell you in a few slides later actually why this retinal is important, right. So, the other difference between rods and cones is as follows. Rods are very sensitive. They can theoretically detect even a single photon of light, and hence they are useful for vision under very dim light conditions. For example, you have both rods and cones in your eyes. So, in a moonless night when you are looking at only starlight is there, that is when whatever vision you have will be because of the rods. Otherwise, cones are used for most of the visual functions, and cones can be used for bright light conditions like sunlight or for detection of color, as I will tell you in the next few slides. So, other than this, the third difference that is between the rods and the cone photoreceptors is that the rod cells are not able to detect flickering light, that is faster than 50 cycles a second, but the cones are able to detect that high frequency of flickering light. Now, let us look at the function of cones in more details, which is the detection of color. So, how does the retina function to detect color? So, what happens is, if you look at again the organization of the retina, you have these photoreceptors which are present in the innermost layer, right. There are three types of cone photoreceptors, and they are called as red, blue and green cones. Now, why are they called as red, blue and green cones? Because they have this conopsin molecule which is slightly different between each of these cone types. Why is it different? Because this makes that cone type able to detect or optimally respond to certain wavelengths of light. Now, all of you might be aware that the white light that we see is made up of different wavelengths of light, and the 7 colors of the rainbow are actually comprising of light of different wavelengths. So, the largest wavelength is the red and the smallest wavelength is blue or violet. So, the same thing happens here. So, these cones have different conopsins. The red conopsin is present in the red cone, the green conopsin is present in the green cone, and the blue cones have blue conopsins, and they can optimally respond to different wavelengths of light. That is, they are maximally stimulated by red light or green light or blue light. Now, how does this translate into detection of color? So, imagine that you are looking at something that is yellow in color. Okay. So, light that is reflected from here will be yellow light, and it will fall on the cone photoreceptors in your retina. Now, yellow light is actually a combination of red and green light. So, yellow light is going to stimulate both the red and the green cones, and the ganglion cells that are connected to the red and green cones are separate, and they send particular processes to the particular area in the brain. So, this is a concept of what is called as a labeled line. So, whatever information is going through the main optic nerve to the brain, the pathways are segregated. So, the brain knows that if some particular wire corresponding to a particular ganglion cell is getting activated, then that must correspond to either the red or the blue or the green cone. So, based on that, color can be perceived. So, here you see when yellow colored object is visualized, then both the red and the green cones are stimulated. So, the brain gets an information that yes, the red line is active and the green line is active as well, right, within the optic nerve. Now, when you look at a blue object, now it will only optimally stimulate the blue cone. So, now the blue line will get activated, and so the brain will interpret that as being blue colored object. So, different combinations of the cones being activated can be coded to represent different colors in the spectrum, right. So, different combinations of red, blue and green, the 3 primary colors can be used to detect every shade that you can think of. Then I would like to move on and talk about another feature of the retina, a structural feature of the retina, which is called as the fovea. Okay. So, if you carefully look at this inner layer of the retina, then in the almost in the center, just above where the blind spot is, where the nerve is coming out, there is a little bit of a depression in the retina. So, this depression is called as the fovea, and it lies at the center of an area which is having yellow colored pigment in the retina. So, this part is called as the macula, right. So, this fovea is like a pit or a depression on the surface of the retina. So, what does it actually look like if you zoom into it and look at it at high magnification? So, this is what the foveal region looks like. It almost looks like somebody put in a finger here and separated out and pushed aside the bipolar cells and the ganglion cells that were connected to these photoreceptors. So, there are several features of the foveal region of the retina. Number one is that it is exclusively consisting of cone photoreceptors. So, there are cone photoreceptors which are packed here at very high density, okay, and that determines the resolution of the image that is formed here. All of you are familiar with camera resolution. You have heard of so much megapixel camera which is better than lower megapixel camera. Why is it better? Because the number of points used to pixels used to create the image is higher. So, the higher the number of pixels used to represent an image, the more is the resolution. So, here you have those pixels correspond to number of cones. So, you have really high number of cones which are packed in over here, and since the cones are better at detecting light in general and they can detect color. So, their foveal region is actually specialized to detect visual detail in whatever you are looking at. So, you can easily test the function of your fovea. Say you have a pen in front of you, this pen has something written on it. Now, if you put the pen here and you look at it sideways without moving your eye towards it, you cannot really see much of what is written on that pen, right. Then you move this towards here right in front of your face. So, that now this is being focused right onto your fovea. So, whatever will be written you will be able to read it clearly. That is because the image that is now being produced is of very high resolution. So, you can resolve very fine differences which you are not able to do here. Here you can basically tell, okay, it is a long shaped object, maybe a pen. Here you can even read what is written on the pen, right. So, that is the function of the fovea. So, this is the region of your eye which has the highest visual acuity. And why does this have the highest visual acuity? Number one reason, it has only cones present there, and these two other layers which were the bipolar cell and the ganglion cell layer, normally light passing through any other part of the eye falling on any other part of the retina would have to go through all these layers. So, there is some loss of light in the process. However, even that minimal loss of light is avoided in the fovea, and the light can directly go into the cones and stimulate them without having to pass through these layers. Does that mean that these cone photoreceptors are not connected to any bipolar and ganglion cells? No, they are. So, these bipolars and these ganglion cells are connected to the cone photoreceptors, but they have just been pushed aside, right. So, then this is what I just described to you. At the posterior pole of the eye, there is a macula which is this yellow pigmented structure. At the center of it, there is a pit called the fovea. The fovea is a thinned out portion of the retina where only the cones are densely packed, hence the high resolution, and it is the point where visual acuity is the greatest, that is the highest resolution image can be formed. So, whenever you want to see detail, you move your eye so that the image is formed exactly on the fovea. Now, I want to move a little bit into the detail of how we perceive light at the level of the rod photoreceptor. So, this is called as phototransduction, which means the conversion of the light stimulus into electrical activity. So, just look at this image and try to figure out how the photoreceptor does this. So, as I told you already, the photoreceptor has this outer segment which is made up of these membranous discs, and these membranous discs are packed with this molecule which is called as rhodopsin. So, rhodopsin is not there alone, it is in combination with retinal, which is derived from vitamin A. So, now you understand the significance of why you get night blindness when you have a deficiency of vitamin A in your diet. That is because you do not get enough retinal. So, your photoreceptors cannot function properly, and you will see in a minute the importance of this retinal. So, when light actually hits the photoreceptor, the outer segment, what happens is this retinal gets converted into another form, it gets isomerized, okay? It changes its structure. And because of this change in structure, it can no longer fit with the rhodopsin molecule. So, it moves out, and now rhodopsin as a result also shrinks and changes its structure. Now, this change in structure of rhodopsin is conveyed to another protein which is also present in the membrane, and this protein, I will not tell you the names because you do not really need to remember the names. This protein can convert another small molecule to its active form. So, this activated small molecule then goes and binds to an ion channel in the membrane. So, this is a sodium ion channel which was normally open. So, in the dark there is always sodium ions flowing into the cell, into the photoreceptor, hence in the dark there is a current which is called as the dark current. Now, what happens when this activated small molecule binds to this ion channel? It closes the ion channel, and then what happens? No more sodium ions are coming into the cell. So, the cell will actually become hyperpolarized. It will become more negative inside than what it was. So, hence this is what is the conversion of light into electrical activity. So, this change in membrane polarity leads to release of neurotransmitter from the end of the photoreceptor, and this excites the next post synaptic cell, which is the bipolar cell, and then passes on to the ganglion cell and to other neurons in the brain. So, this in a very short and nutshell fashion I have described to you what is phototransduction. Now, we are going to move on to the next sense, which is very important. This is the sense of hearing, and the sense of hearing all of you are aware of is through the sense organ which is called as the ear. So, this is a diagram that shows you the different parts of the ear, and we will go through each one of them. So, in a broad sense, the ear is divided into what is known as an external ear, which extends up to here, then what is known as the middle ear, which is just this red colored object here, which is colored in red, this chamber. Then the inner ear, which is this part, that is the last ear, important part of the ear. So, let us talk about the outer ear first. So, the outer ear composes of the pinna, which all of you can see. So, this is the pinna, and then there is a ear canal, which is given a very fancy name called external auditory meatus or external auditory canal, right. So, this is nothing but the ear canal. So, most of you are only aware that it exists, none of you have ever been able to see what lies beyond. So, you probably know that at the end of the ear canal, it is like a tube which ends in a membrane. So, there is a membrane which covers the end of this tube, and this membrane is called the tympanic membrane, or in very simple terms the eardrum. Okay. There are very, very fine hairs that are present in the surface of the inner surface of this external auditory canal and on the surface of the pinna. The function of the pinna is to collect the sound waves and direct them into the ear canal, and not only that, the function of the ear canal is to conduct the sound waves into the so that they hit the tympanum, but also the hairs and a lot of wax that is present in the ear canal traps many microorganisms and dust, which would otherwise hinder the process of detection of sound. So, then let us look at the middle ear. So, as I told you, the chamber that lies beyond the tympanic membrane or the eardrum is called as the middle ear. So, this chamber has 3 small bones which are called as ear ossicles. So, the bones have very fancy names like malleus, incus and stapes. What they are called in very layman's terms is the hammer, the anvil and the stirrups. So, why are they named like this? The reason is because of their shape. So, the malleus looks like a hammer and the anvil is the structure on which the hammer is struck, right. So, the anvil, the incus looks like the anvil. So, here is the malleus and here is the incus, and the third bone looks like this horseshoe shaped thing. So, this is actually like the stirrups. So, if you are familiar with horse riding, you will know what these stirrups are. So, when you sit on the saddle, then you put your feet into these structures which look like this. So, your feet goes in there and these are called as the stirrups, right. So, that is why this structure is named as the stirrups. Now, these are three bones and the malleus part is connected to the tympanic membrane. The stapes is on the other hand connected to what is known as a oval window, which I will refer to later. So, this is the border between the middle ear and the inner ear, right. So, the ear ossicles are there so that they increase the efficiency of transmission of sound. So, when the tympanic membrane will vibrate due to the sound waves hitting it, the bones will also vibrate along with them, then conducts the movement to the inner ear, which is starting here at the oval window. And I will tell you in a minute how this leads to the production or detection of sound by the inner ear. Another important fact that you need to remember is that the middle ear is a closed chamber, but it has to be open to the outside. So, this is the opening to the outside which opens out into your nasal cavity through the pharynx, throat and nasal cavity. This is called as the eustachian tube. So, the function of the eustachian tube is to equalize the pressure between the external ear and the middle ear, because otherwise there would be too much pressure build up there. So, let us then move on to the third part of the ear, which is the inner ear. So, the inner ear is actually a very complex structure. So, you see this coiled structure, this is called as the cochlea. So, the whole thing is actually embedded into your skull bone. So, it has a bony part and it has a membranous part. So, the bony part is actually the casing of this. So, the whole structure is called a labyrinth. What does a labyrinth mean? Labyrinth means a winding path. Okay. So, because of this winded structure of this thing, it is called as a labyrinth. So, you have a bony labyrinth which is the covering, and then inside it you have a membrane. So, what you have really is a hollow tube which has been rolled up like this. So, this is what the cochlea looks like, right. So, this bony labyrinth has a series of channels and there is a fluid that bathes it, which is called as the perilymph, and then there is a membranous labyrinth, which is this tube which is made up of a membrane, a hollow membrane bound tube, and this tube is rolled up like this, like the snail. You have seen a snail, the shell of the snail, it looks like that, and this is called as the cochlea. This is the most important part of the inner ear, which is used for detection of sound waves. Now, the cochlea, if we look into it, it looks somewhat like this. As I told you, it is a tube. So, imagine it is a hollow tube which I have now unrolled and I have opened it up like this, right. Now, this hollow tube is divided into three chambers. Actually, there is a upper chamber, there is a small tiny middle chamber and there is a lower chamber. So, this is actually a cross section of the cochlea. So, now you can see that the entire section of the tube is divided into three chambers, 1, 2 and 3. The upper chamber is called the scala vestibuli, the middle chamber is called the scala media and the lower chamber is called the scala tympani. So, here is the scala vestibuli, here is the scala media and here is the scala tympani, right. Now, this is a opening here on which the stapes actually sits. So, this is a membrane covered opening and this is called as the oval window. And then there is another opening here which is also covered by the membrane, which is called as the round window. Okay. So, there are two membranes which actually divide this entire space into three compartments. So, one is called as the Reissner's membrane and the other is called as the basilar membrane. So, the basilar membrane is this part which lies at the bottom of the scala media, and this is the important part that we are going to discuss in the next slides. So, this is again filled with a fluid which is called as the endolymph. Now, what happens we will discuss in the next slide. So, if I tell you that the basilar membrane is a very thin membrane that is going along the entire length of the tube, right, the cochlear tube. But the basilar membrane actually, if you represent it like this, it is a membrane that is shown in a single line. So, for simplicity's sake I am not showing the three chambers, but I am just showing you the membrane, the basilar membrane which is moving along through the entire length of the cochlea, which has been opened up like this. So, what happens is when sound waves are coming and hitting the oval window, the stapes will be vibrating according to the alternating rarefication and compression of air, which is what is a sound wave. So, then the fluid here that is filled up will have a wave that is generated. So, this wave will cause the membrane to move up and down. So, the basilar membrane will do this and leads to the separation of different frequencies of sound. Why? Because if you take a look at the basilar membrane just by itself, it looks very different from one end of the cochlea to the other. So, from the end towards the windows, it is very thin and it is thick, and here it becomes very, very thin and very, very stretched and floppy. So, that immediately tells you high frequency sounds will make this part of the membrane vibrate more and low frequency sounds will make this part of the membrane vibrate more. So, already you can have a separation of different frequencies of sound on different parts of the basilar membrane. Now, the basilar membrane generates a wave which causes this travelling wave to go along this entire basilar membrane, and the highest displacement of this wave happens here if the frequency of sound is highest and here if the frequency of sound is lower. So, now let us see what happens in terms of how this sound wave is converted into electrical activity. Okay. So, this is done by what is known as the organ of Corti. So, on the basilar membrane surface there are some cells which are organized into this organ of Corti. These cells are known as hair cells. So, these hair cells are very unusual looking. They are very small and they do not look like a neuron in any way, but they have this very long projections of their membrane, finger like projections of their membrane on the surface. So, these are called as stereocilia. Okay. So, these stereocilia are all connected to a membrane on top, which is called as the tectorial membrane, right, and then the basilar membrane is at the bottom. So, the hair cell is actually spanning the tectorial membrane and the basilar membrane. The tectorial membrane is a rigid structure and it cannot move in either direction, but the basilar membrane is going up and down. So, what happens when the basilar membrane goes down? Because the hair cell cannot change its shape much. So, in order to fit into a lesser space, the stereocilia will bend like this in this direction, and when the movement of the basilar membrane is going down, then it will become straight once again, right. So, this movement, mechanical movement of the stereocilia on top of the hair cell is what will generate an electrical impulse from this cell. So, this we will see in the next slide. So, this is an example by which there is a conversion of a mechanical stimulus to electrical activity, and how this can be done is shown in this series of images. What you can see here is a membrane of a cell in which an ion channel is embedded, and this ion channel is not open. it is closed by this spring like this lid which has a spring attached to it. Now, if you stretch this spring by some mechanical means, you can open up the lid and you can allow ions to go in. This is one way that mechanical forces can lead to opening of ion channels. So, now you understand how mechanical forces can lead to electrical activity of a particular cell. So, to explain this in even more details, let me draw what the top part of the hair cell looks like. So, you have the stereocilia like this, right. So, when the tectorial membrane is attached to the top, what happens is when there is a displacement of the stereocilia in this direction, there are some ion channels which are present on the membrane, but they are closed and they have a spring like structure attached to the next stereocilia. So, when there is a displacement in this direction, you can imagine what happens, this lid will open up and now ions can come in, and this will lead to depolarization of this cell, and this will eventually lead to release of a synaptic neurotransmitter at the bottom of this cell, and then activity of the next neuron in the series. So, this is how the auditory signal, which is sound waves, is converted into electrical activity of a cell. Now, before I move on and finish up with the ear, let me tell you about another organ which is also present in the inner ear. This is called as the vestibular apparatus. So, most of you are aware that if you turn around multiple times, you do not usually fall down. There is something that tells you that restores your balance. The reason you can still stand and not fall down immediately is because of this vestibular apparatus, which is useful for maintaining your balance. So, what does this vestibular apparatus comprise of? It is made up of 3 canals. So, there are these 3 loops of tubes called as 3 semicircular canals, okay, and they are filled with fluid just like the cochlea, right. Then it has a basal part which comprises of 2 units. One is called the utricle and the other is called as the saccule, and these are the 3 semicircular canals which are all oriented in along the 3 different axes, the vertical, the horizontal and another one. So, these membranous canals are suspended in perilymph and they also have a fluid inside, as I told you. And within this, the inner portion of this, if you look at the magnified view inside, there is fluid flowing and there is this structure which has connection to the hair cell, which is present at the bottom. Okay. So, there is this crista ampullaris which is present here, and this has hair cells embedded in it, and this structure is spanning the entire width of the semicircular canal. So, when there is any displacement, so what does this whole apparatus do? The semicircular canals can detect head rotation movements, which is movement of your head from side to side, and the 2 otolith organs that I just told you, the saccule and the utricle, they sense linear motion. So, when you are moving in a straight line. Okay. So, how do they detect? The fluid moves. So, this whole structure moves, and similar to the detection of sound waves, the hair cell gets activated here as well with this movement. Okay. And this whole system is then responsible for maintenance of the balance of the body and the posture, because it always gives you feedback on what is the position of your body with respect to your surroundings and where is your head positioned with respect to the rest of your body. Okay. So, before I end this lecture, I wanted to tell you a little bit more about the 3 other senses that we have not touched on in much detail. So, the first of this is what is known as the somatic sense, which is the sense comprising of various different components, which is touch, proprioception, pain and temperature. So, these are all grouped together as the somatic senses, and the sense organ that is used to detect it is the skin. So, your skin has many endings of nerves, sensory nerves, which have certain organs at their end, a structure of a lot of membrane wrapped around. This is the one of the nerve endings, okay. And this, imagine this is a like lot of, it is covered by layers and layers of membrane. So, what this allows you to do is, if this is present near the surface of your skin and there is a touch stimulus here, this causes a deflection of the membrane over here, right, and this is conveyed to the membrane of the nerve ending. Now, the membrane of the nerve ending is stretched because of this touch, because there is a pressure being applied here, right. So, this stretch of the membrane can lead to the opening of an ion channel which was otherwise closed. And then in the previous part I also talked about how other kind of gates can be there on the ion channel which can be opened by stretch. So, sometimes the ion channel is connected to the membrane through these kind of structures, and when there is a stretch or a physical displacement, then the spring lengthens and this snaps open the lid as though on the channel, and then the ions can go in. And as long as ions can go into the cell, it can change the membrane potential, and that leads to the formation of a new nerve impulse. Now, this is the same mechanism that is operational for detecting touch, with which you can actually perceive the shape and size of objects and the surface texture of objects. Then proprioception, which is these same nerve endings are present deep within your muscles and your joints, which tells you the pressure that is there on each joint at any given position. So, you are not aware of it, but your brain knows that how much pressure is being applied. For example, when I am standing or when I am sitting or when I am holding my hand in a particular position or my leg in a particular position, what is the pressure in each joint. So, that is sensed through this proprioception that I am talking about. Then there are free nerve endings which can sense pain. So, pain is usually sensed through chemicals that are there when there is some kind of a cut or an injury or some kind of an infection that leads to production of various stimulants which can stimulate the pain receptor. And temperature has a different mechanism for sensation, where you have a change in temperature can activate the nerve endings of specialized nerves which are separate for cold sensory nerves and heat sensory nerves. So, this is in total a summary of how the somatic senses are functioning. Now, let us talk about the two chemical senses, the first of which is called olfaction or the sense of smell. So, in the nose, what you have is, if you look inside, on the roof of your nose you have this sensory epithelium which has many cells embedded in here. So, if you look at a blow up of this, you can see that these are very specialized kind of cells which have some again extensions which are called as cilia. So, these cilia contain some specialized kind of ion channels embedded in there, or certain kinds of receptors embedded in there, proteins which can bind to certain chemicals. So, each kind of cell has a particular protein which can detect the presence of a particular chemical. The only criteria is this chemical has to be volatile, so that it can enter into your nose and it can interact with this particular protein which is present on the surface of this cell, right. Once this binds, this is an example of a ligand gated ion channel. So, very often this binding leads to opening of an ion channel, either directly or indirectly it activates another protein which leads to opening of another ion channel, and then again positively charged ions can come in and this can depolarize this cell, and then this cell has an axon which goes on to the part of the brain that is right above your nose, which is called as the olfactory bulb, and this stimulates the olfactory neurons which are present there and then conveys this information to other parts of your brain. So, you can detect the presence of a chemical in your nose and convey this information right up to your brain, and this is what comprises your sense of smell. Very similar to this is what is known as the sense of taste or gustation. The sensory organ is different in this case, which is the tongue. So, the tongue looks like this, and it has certain regions of the tongue are specialized with certain kinds of cells embedded in them. So, on the surface of the tongue you will see there are some depressions, and these depressions have a very specialized kind of cells located here, which are called as the taste bud. So, this is a taste bud. So, this taste bud in high magnification looks like this. So, there will be a few cells that are present here. So, again like the olfactory sensory neurons, these taste bud cells have certain receptors present on their surface. So, these taste bud receptors can actually bind to certain molecules that are the tastant molecules, and therefore, depending on what kind of a taste bud it is, it can detect various tastes such as salt, sugar, bitter, sour, and there is another one called umami, which is the meaty taste. So, there are different receptors, and individual taste buds will have each of these cells containing different receptors that can optimally detect each of these tastants. So, when the tastants bind to the receptor, this again leads to opening of an ion channel which can depolarize the cell, and this is then connected to a nerve which is called as the gustatory nerve, which goes and takes this information to the part of the brain that is devoted to perceiving taste. So, this is how the two chemical senses, taste and olfaction, function. So, with this we are going to end today's lecture, and before I go, let us discuss certain questions that can have arisen in your mind. So, the first question is, how can the ear distinguish between different frequencies of sound? So, I kind of explained this in the lecture. What happens is, if you open up the cochlea, you can see that it has a basilar membrane, right. So, this basilar membrane has different mechanical properties along its length. So, here it is very taut and it is thick, and here at the other end it is very floppy and it is very wide. So, high frequencies of sound can optimally stimulate the membrane here. So, make the waves really amplitude here, whereas here this part of the membrane will be stimulated by low frequency of sound waves, right. So, only when the membrane is moving up and down or undulating at a certain rate is it capable of stimulating hair cells. So, hair cells are sitting on all parts of the basilar membrane. So, when the high frequency of sound comes, the membrane displacement is sufficient here to activate the hair cells over here, and only when it is low frequency it can optimally activate the hair cells over here, and each of these hair cells are connected to other neurons which are the output from these. So, when these neurons are stimulated, that means high frequency sound is coming, and again a label line kind of concept operates here. Just like you have different wavelengths of light being detected by photoreceptors, you have different wavelengths of or frequencies of sound being detected by different neurons within across the entire breadth of this length of the basilar membrane. So, now you can understand how the ear can distinguish between different frequencies of sound. Then let us talk about the second question. What are the features of the fovea that make it the region of the retina with the highest visual acuity? So, remember the fovea, this was the part of the retina which had this pit like depression. So, what are the features that make it have highest visual acuity? Number 1 is this pit. So, the bipolar cells and the ganglion cells have been moved aside, so that light can directly hit the photoreceptors. Second point is that only cone photoreceptors at a very high density are present here. So, because of this, the highest resolution image is produced at the fovea, and hence you call it the center of highest visual acuity. The third question is, how is sound transduced into electrical impulses in the hair cell? So, here I told you, if you look at the hair cell structure, then it has the stereocilia, and then the stereocilia have on their tips certain ion channels. So, if you look at a high magnified view of the end of the stereocilia, you can actually see that there are certain ion channels present here which are normally closed, and there is a spring like structure attached to the membrane of the next stereocilia. So, what happens is when there is a displacement of these stereocilia because of the hair cell moving up towards the tectorial membrane. So, this displacement will lead to a stretch in the spring, and this mechanical stretch will make the lid of the ion channel fly open, and then this will lead to ions that can come inside the cell and depolarize the hair cell. So, mechanical sound waves are transduced into electrical impulses. The fourth and the last question is, what are the differences between rods and cone photoreceptors? So, this is quite simple, you can get it from the name itself. The first difference is in the shape of what is called as the outer segment. So, the shape of the outer segment is rod like in rod photoreceptors and is cone like in cone photoreceptors. Now, these have membranous discs which contains two different pigments. So, in case of the cone cells, it is the cone opsin, and in case of rod cells, it is rod opsin. Now, cone opsins are of different kinds, red, blue and green. So, you can detect 3 different colors, and rhodopsin however can respond to all the different wavelengths of light, but it is much more sensitive. Very little light is needed to stimulate it, to activate it. So, this rhodopsin therefore can theoretically detect even a single photon of light. But the cones are better in one other aspect, that is, if you have flickering light or changing light, then anything higher than 50 hertz cannot be detected by rods, but cones are good at detecting that. So, with this we end today's lecture, and I thank you all for listening. I hope you enjoyed learning about the sensory systems.